It's an honor for me to be here today, and thanks to Shiva for inviting me to participate. And I also want to thank all my investigators, because it's thanks to them that I'm here today presenting the trial. So today what I will do is present the trial, but first tell you why we decided to do this trial. So by now, I think we should all know that postpartum hemorrhage is the main cause for maternal mortality. That WHO recommends to use uterotonics during the third stage of labor for preventing PPH, and that oxytocin is the drug of choice for third stage of labor. Oxytocin is a very good drug. However, if we follow the label, we know that we have to keep it in the fridge. It's a drug that has to be stored between 2 and 8 degrees. Well, and depending on the manufacturers, there are others recommend to store it below 25 degrees. Anyhow, we did a systematic review and we looked at the quality of oxytocin in Africa, and we found that the quality of oxytocin is low nowadays. And there are several causes, being the main reason the cold storage. For many settings, it's very difficult to store the drug in the fridge, but also not only at a health center, also along the supply chain, it's very difficult to keep the drug between 2 to 8 degrees. So there is a huge need for a heat-stable uterotonic. There are people working nowadays with misoprostol, with oxytocin inhaled, and we decided to work with carbetocin room temperature stable. So why do we think that carbetocin is important? Carbetocin is an oxytocin analog. It's produced by Ferrin Pharmaceuticals. This product is very much like oxytocin, but it's more stable molecule. It's resistant to several enzymes for which oxytocin is not resistant, and it's also stable to several chemical reactions for which oxytocin is not. All these characteristics makes carbetocin to have a longer half-life, and therefore induce a prolonged uterine contraction. And last but not least, carbetocin now, we have formed it's room temperature stable. So Ferrin Pharmaceuticals were able to modify the molecule, change the excipients. Now the excipients are part of the GRAS, what we call GRAS, generally recognized as safe excipients, and thanks to them, the carbetocin now can be kept out of the fridge, which is, I think, huge, because it will really answer our needs. In addition, carbetocin is a product that has been marketed for the last years now. It has been first registered and approved in Canada in 1997, and now it's registered in 70 countries for the prevention of PPH in cesarean deliveries, and in 9 countries for the prevention of PPH in vaginal deliveries. All this gives the advantage that now we know the safety profile of carbetocin, and we can say that it's very similar to the oxytocin. Side effects are similar to oxytocin. We have nausea, we have feeling of warmth, chills, pain, headache, hypotension. But they're really at the same level as oxytocin. So someone should say, well, then start using carbetocin. But no, because if we look at the evidence now, we look at there is a Cochrane systematic review published by Lim Su in 2012 that they look at the evidence on carbetocin for preventing PPH. They found 11 studies, but these studies were small. The total sample size of these 11 studies is only 2,600 women. Most of the studies had the indication of cesarean deliveries and IV route. And there is little data on the endpoints such as blood loss of 1,000 mils or maternal morbidity. So the conclusion of the systematic review is that there is need for further research to look really if carbetocin is effective in preventing PPH in vaginal deliveries. So here is where we come. This is our trial. It's a phase 3 randomized. It's double-blind. It's active control because we are comparing carbetocin RTS with oxytocin. It's multinational, and it's non-inferior. And we are looking in vaginal deliveries. So in this slide, what I wanted to show is the complexity from the managerial point of view. We are used to pragmatic trials studies from WHO, but because this trial, we will use the data to register the drug, the carbetocin room temperature stable in many countries, then we need to follow some rules. So we have our partners. We have Merck for Mothers, who is funding our project, but also giving technical expertise. We also have Ferring, who is producing the Carbetocin RTS and has the know-how of the product, so is helping us with the technical expertise too. We have Concept Foundation, who will come once the trial is finished. And all these big partners, we are all together governed by an agreement. So there is a very important agreement signed with all the partners in which we specify the roles and the responsibilities of each of us, but we also say our main objective. The main objective of this trial, from the scientific point of view, is to see if Carbetocin RTS is as effective as oxytocin. But also, our objective is if Carbetocin shows to be non-inferior to oxytocin, then we will make the drug available in low-resource settings at an affordable price. Nowadays, Carbetocin is a very expensive drug, so with this agreement, we are ensuring that we will have Carbetocin RTS in the public sector of low-resource settings at a price similar to oxytocin. This is around 25 cents, 30 cents, which is a huge difference in price. We also have other partners. We have Quintiles, who are helping us with monitoring and regulatory processes in all the countries. We have Iberslev, it's a packaging company. We have Excellent Fixable Drapes, providing us the plastic drapes to measure the blood loss. And we have our center, a collaborating center in Argentina, doing the data management. So, in this map, I'm showing the countries participating. There are 10 countries participating: Argentina, Nigeria, South Africa, Egypt, Uganda, Kenya, UK, India, Thailand, and Singapore. The total sample size is 30,000 women delivering vaginally, and the recruitment period is 12 months in each of the countries. So we have two primary objectives. One objective is to look at 500 ml or use of additional deuterotonics, and this objective is for regulatory purposes. We have another primary objective, this is our objective, to look at the effectiveness in preventing severe PPH, 1,000 ml. And this is needed if we want to include then the results in our WHO PPH guidelines and make a submission to the essential medicines list. So, as I said, we are going to include women delivering vaginally with singleton pregnancies, that they are not distressed, and that they have a cervical dilatation of less than 6 centimeters. We are going to exclude all those women who have a planned cesarean section, with allergies, with serious cardiovascular disorders, hepatic or renal disease, and here in India it's going to be slightly different. The serious will not be there, so we will include any cardiovascular, hepatic or renal disease will be excluded, and epilepsy. As you see, depending on, in each country the situation is slightly different, and we have to follow what our regulatory authorities request. So the protocol is slightly changed, for example, in this case. So this is intervention, it's double blind, so these are the ampoules, they look all the same, but half would be Carbetocin RTS 100 micrograms, and the other half would be oxytocin 10 international units, which is the comparator drug. So the intervention would be administered immediately after birth, within 1 minute, and then once the cord is clamped and cut, we will place the plastic tray to measure the blood loss for 1 hour, and if the woman continues to bleed, for an extra hour, total 2 hours. So our primary endpoints would be blood loss of 500 ml or additional deuterotonics, and blood loss of 1,000 ml. Our secondary endpoints would be blood transfusion, manual removal of placenta, additional surgical procedures, serious adverse events, severe morbidity, maternal death, and the composite outcome, which is severe morbidity and maternal death. There are others, but these are the main ones that we are looking at. So when are we going to say, okay, we finished the trial? For the women, when the woman is discharged, or if she's transferred to a higher level of care. For the whole trial, when we lock the database, so we have to clean and then lock the database. That's our end of the trial. So what would happen if Carbetocin RTS shows to be non-inferior to oxytocin? Hopefully it will. We will include the results in our WHO PPH guidelines, and we will recommend Carbetocin RTS in those countries where they don't have a fridge or they cannot ensure the cold chain. We will make a submission to the Essentials Medicines list to include the product in the Essentials Medicines list. And the most important thing, but I think it's a huge issue, we will make sure that the price will be as low as it is oxytocin nowadays. And this is, I think, the beauty of the trial, that thanks to the intervention of WHO, you can convince the pharmaceutical company to reduce the price. I mean, we are working a lot, and this represents big efforts from all the parties, but we will be there. So thank you very much, and thanks to all the investigators really for participating in this.
NEJM and the Lancet. He serves or has in the past served on several international and national committees, including the BJOG Editorial Board and the RCOG Wellbeing of Women Research Panel, various WHO committees, the Early Pregnancy Clinical Studies Group, and the Reproductive Medicine Clinical Studies Group. He is the founding trustee of AMALIFE, a UK registered